Where we left off last time, we were talking about lipids and lipid structure, and specifically these non-traditional esters, which we are terming glycerides. These glycerides came in two forms: neutral glycerides versus charged glycerides. Our neutral glycerides were, as expected, neutral. They had no residual charge on them. An example here is of a triglyceride. They had no charge whatsoever on it. Charged glycerides will have charges on them, or do have charges on them, but we find that neutral glycerides are primarily used for energy storage, and that's what we looked at last time, in which these triglycerides mainly are held within these adipose cells, adipose tissue, which is essentially fat tissue, which can then be accessed by animal systems if an amount of energy is ultimately required there. So that was where we were looking at for neutral glycerides. Now, charged glycerides, we had looked at before. We had made the statement that charged glycerides are primarily used as cell membrane building blocks, much different than the neutral glycerides. Here over on the right-hand side is a representative charged glyceride. We have on it certainly some charged system over here. This phosphoester and the small molecule attachment here, which in this case is ethanolamine, in which we have at physiological pH, any amines we see here are positively charged, and any oxygens on a phosphate are negatively charged. So this is the charged portion, if you will, of a charged glyceride. There's also, as we're going to see, an uncharged portion, and we'll talk more about that. That's essentially a diglyceride, a neutral diglyceride, which has on its third hydroxyl group an attachment, a covalent attachment to this phosphate and its subsequent substitution. More to the point on these charged glycerides, last time we had looked at three representative ones. We have here phosphatidylethanolamine, phosphatidylcholine, and phosphatidylserine. And these three types of structures can generally be brought into one type if we look at them. And I'll put up here on the upper right-hand side, can be generally thought of in this sort of sense, where we have a general charged head group here, and two fatty acid tails over here, and we have that connection through the traditional glycerol structure. So that would be what would be we would consider a general structure of all three of these particular cell building block components. They are written, I guess, suggestively as phospholipids. They're referred to in general as what are called phospholipids because it is a lipid, a diglyceride, covalently attached through a phosphate group, which is itself substituted with a charged head group. So these phospholipids, as we see here, all three of them are the major components in virtually all cell membranes. In cell membrane, we're going to see how that cell membranes get put together down the road towards the end of this unit, but they're the major component in cell membranes, one of these three types of building blocks which we see here. Now, there is one other type of cell membrane building component, and it's a very specialized one, and it's for a very specialized type of cell. Whereas all three of these phospholipids here are in virtually any cell membrane within our body. They make up the cell membrane, whether it's skin cells, with fat cells, or any other types of membrane we see in there. Nerve cells have a different type of structure and therefore require a different type of building block. And the name of this building blocks are known as sphingolipids. A sphingolipid is the major cell membrane building block for specifically nerve cells, for nerve cell structure. And while similar in structure to our general phospholipids, sphingolipids have three notable differences, three of them. If we look at the standard 3-carbon connection, there's that 3-carbon connection we normally see in any sort of glyceride. Here we see it up here in the upper right-hand side as well. There are differences with the connection, if you will, to that 3-carbon unit. So let's go back down and look at our sphingolipid again, and we'll note three specific differences within there. First one is that we have a direct connection on one of our fatty acid tails, or one of our hydrocarbon tails, to that 3-carbon unit right there. It's a direct carbon-carbon bond connection, which we see that. Now, as such, this surrogate, if you will, for a glycerol does not utilize its hydroxyl in an ester linkage. So we also have, as a result of that direct connection to the 3-carbon unit, a free hydroxyl just hanging off there, which we don't see in the phospholipids. So there are two related differences between sphingolipids and phospholipids. The third major one, if you look here on our sphingolipid, is that in the central carbon connectivity, what we have here, let's see, this would be an amide linkage. That is an amide. Now, normally we have an ester linkage in a standard phospholipid. That's an ester, but down on the sphingolipid, that ester linkage has been converted into an amide linkage. So let's start thinking about the structure of these biological membranes. And by that, I essentially mean cell membranes, how they actually come together. And to first do that, we have to look at the structure of a standard phosphoglyceride. It's a regular phosphoglyceride. In this case, it's phosphatidylethanolamine. And we see sort of an echo of something that we have looked at before, in that we have in this phosphoglyceride, it is a conflicted molecule with respect to how it likes to deal with water or an aqueous environment. We have a hydrophilic portion. This hydrophilic portion on any phosphoglyceride, hydrophilic means water-loving. Now notice it's water-loving because it has these charged areas right here. Charges love water, and it also has frequently, most of the time, hydrogen bonding, in this case from that amine right there. So that's very much like water, like dissolves like. So this is considered a hydrophilic portion of the molecule. This is all covalently bonded together. Now, covalently bonded to that hydrophilic portion, we have a portion which is hydrophobic, water-hating, meaning there is no there's no hydrogen bonding. It's mostly hydrocarbon, which is very, very different than water, and dissolves unlike. So therefore, we have this hydrophobic portion on the same molecule as well. Now, as we've shown before, or as I've talked about before, these very complex structures in biochemistry are frequently brought down to the level of the cartoon. And we saw this before in another case. And in this case, we have over on the right-hand side this cartoon version of how we're from now on really on a large scale going to be expressing these phosphoglycerides or phospholipids, in which we have a sort of this spherical piece right here. That is considered the hydrophilic portion, or it is representative, a cartoon representation of the hydrophilic portion, and these two sort of tadpole tails coming off of it, that is representative of the hydrophobic portion. So going forward, we're going to be referring to these phosphoglycerides as this sort of two-tailed tadpole, if you will. Now, this is very reminiscent of when we were talking about soap micelles and how the fatty acid carboxylase phospholates aggregate into a micelle way back in unit 5, I think it was, and we had to go to the level of the cartoon as well on that. The cartoon didn't exactly look like what we see over here on the right. I think it was basically a spherical ball and only one tadpole tail, but you get the idea. We had to revert to the level of the cartoon. So with this cartoon in mind, we can make the statement now that when these phosphoglycerides are introduced into an aqueous environment, basically they're put into water. And since animal systems or biological systems are mostly water, when these phosphoglycerides are expressed into the aqueous system, they spontaneously aggregate into a structure which is essentially a membrane. It suits the hydrophilic and hydrophobic structures of their makeup. Remember those soap micelles? These micelles had the hydrophilic portion on the outside and the hydrophobic portions pointing to the inside, so they wouldn't have to come into contact with water, whereas the hydrophilic portion did come into contact with water. A very analogous sort of thing goes on with these phosphoglycerides. However, they don't form micelles necessarily, but they are more induced to spontaneously form membranes. And let's take a look at how that happens. So how might these phospholipids spontaneously aggregate themselves such that their hydrophilic portions can come in contact with the aqueous environment of biological systems, and the hydrophobic portions can avoid any aqueous coming into contact with any aqueous atmosphere, if you will. So what happens is, rather than going into a micelle, they come up with a sort of situation where the hydrophilic charged groups are on each side of the membrane are associating with the aqueous environment here and down here. Now, the hydrophobic hydrocarbon tails are pointing inward in one direction on one area and inward in the other direction in the other area. What we're looking at here is a cutaway. It's a cross section, and if you can imagine sort of this moving out from each other sort of in a planar surface, we have the basis of what is known as a membrane. And if we look closely at this, and again, we're showing this to be just a tiny cutaway of this area, we have plenty of water on both sides. And don't those hydrophilic portions of the phosphoglycerides associating together love that water? And on the other side, we have water as well. So we have water on one side and water on the other side. Now look what happens on the inside of this membrane. It completely excludes water throughout. No water is allowed in here. There's no water allowed whatsoever. So now a water molecule, if you will, let me clean this up a little bit. If I'm a water molecule over here on the other side, I cannot go directly through and be a water molecule on the other side. I simply can't do that. However, since we're looking at just a small cutaway of this membrane, of this impermeable surface towards water anyways, we could quite easily say, all right, well, if my water molecule is here, I'll just go find the end of the membrane and go around. And there, I've gone from one side of the membrane to the other. So the issue here, or the solution to this, if a membrane is truly going to be impermeable to water, is to make it such that there is no end of the membrane where a water molecule could perhaps go from one side of the membrane to the other. So let's see how that might be accomplished. Try to imagine now this sort of membrane, this planar membrane extending out in all directions. What if it collapsed upon itself? This planar membrane sort of sort of formed into a sphere. And here is a cutaway that we show here in this view of a sphere. We still have this lipid bilayer membrane, lipid bilayer, because it's made of lipid fat, and it's two layers, bilayer. We still have that anywhere we go around on this. Remember, this is a cutaway of a sphere. We have that so that any water molecule that is on the outside here, this is a watery area out here, cannot get to the inside. It simply cannot, like we did before, or as we showed before. Notice now that we have, because we've taken this membrane and we've enveloped it upon itself into a sphere, we have a watery outward, water all around, if you will, and also a watery inner compartment. However, no water from the inside, from the inside of this compartment can get out to the outside, and no water from the outside can get into the inside. So what we have is now a true impermeable membrane, at least for water. Now that's important because all nutrients and oxygen and everything that is carried throughout biological systems are virtually all aqueous based, meaning they have to be able to dissolve in water. So if this was our representative cell membrane right here, it would be very difficult, as we said, or impossible for water to get through through that lipid bilayer. It will be forcefully pushed out if it tries to get through. But that can be a problem now if that water is carrying vital nutrients dissolved in it, dissolved gases, other things that need to be brought into the inside of the cell. Now, a cell is a living organism, and as such, it requires a constant influx of nutrients and oxygen, a constant outflow of waste products and carbon dioxide and that sort of thing. So we're going to have to augment this model somewhat in order to allow it to, in a controlled fashion, allow nutrients and oxygen and that sort of thing through the membrane. But let's take a larger view, if you will. What we have here is just a planar cutaway of this lipid bilayer, of this membrane. Let's look at it more of a three-dimensional type of way. So here now is a much better way than I could express in an art form. Here we see a much larger, we're stepping back by a certain amount. We see essentially just a model, really, of how a cell membrane actually works. Here we have water outside. We also have water inside. And this, again, it's a cutaway. Obviously, this wouldn't exist in real life. But in all cases, we have this lipid bilayer excluding water from going from the inside to the outside, as well as from the outside to the inside. As I said in the last slide, that can be a problem. That can actually be a problem because we're going to have to be able to get the components that are dissolved in the water through that membrane. Let's see how that might be able to happen. Let's look at a perhaps more realistic depiction of what this membrane looks like in an actual cell. As we look at this view right here, we do indeed see the cell membrane, that lipid bilayer with its little tadpole tails sticking inward into the layer, but we see a lot more stuff within there. Inside the membranes, we see the cutaway of the membrane. Within it, it is really a fluid sort of environment. It's fluidic. If you had to say, well, what does that mean? What does it mean to be fluid inside a membrane? It would have the consistency of, say, olive oil. That is a known fluid, and we know its viscosity and that sort of thing. So it's sort of oily in there. It's somewhat fluid, and because of that, what can float within this membrane are proteins. Proteins can, as we we see here in these depictions literally float within the membrane like sort of a ship being supported by water going through the ocean. And these proteins can float in the membranes. They're called transmembrane proteins, and what they can do now is to allow for the influx and efflux of materials through that membrane. It is absolutely required, because what we have here, we have the outside of the cell and we have the inside of the cell, and both of those are aqueous environments. Because the cell is a living thing, it requires an influx, as I said, of oxygen and nutrients and that sort of thing, and has to be allowed to cast out any waste products, carbon dioxide, ions, and that sort of thing. The real interesting thing about these transmembrane proteins, for instance, this one right here, it's known as a channel protein. And a channel protein, as you'd think, has a channel going through it. It's like a tunnel going through it. However, that channel is very, very specific for a certain nutrient or something that the cell ultimately needs, and it allows those ions or nutrients or oxygen or that sort of thing to go through that channel. It's a very, very controlled way of delivering or bringing out one particular nutrient or waste product or dissolved gas or something like that, so we don't have this this willy-nilly influx and efflux of materials. It is very exquisitely controlled. Within here, so this channel protein, let's see, right here might deliver specifically sodium ion or something like that, and that's the only thing it would ever deliver. It excludes everything else. This channel protein over here might be specific for allowing the efflux of waste products such as carbon dioxide or that sort of thing, but it's specific only for that. So we have this controlled ability to allow the influx and efflux only of what is necessary for the survival of the cell and the internal organelles of the cell. Along with actual nutrients and waste products and deliveries to and fro through the membrane, we also have we need the ability of signaling. If something is going on outside of the cell and that needs to be made aware of the inside of to the inside of the cell, we have signaling proteins as well. And here would be an example of a signaling protein right here. And what that can do is it can allow for a single signal transduction through that membrane. For instance, if a molecule were to dock with this part of the peripheral protein that is on the outside of the cell, that might allow for a conformational change, which would release this piece into the interior of the cell, and that would then dock with some signaling system, which would be able to deliver a message from the outside of the cell towards the inside of the cell. Because remember, information in the case of going through this membrane is just as important as nutrients or waste products and that sort of thing. Here is just another rendition of the exact same thing we looked at before. Here we see now is a cutaway of that lipid bilayer, that membrane, that cellular membrane, and we have within it, floating within that fluidic sort of medium within the interior of that membrane, we have these transmembrane proteins. So here is a representative channel protein which can deliver, say, some oxygen or some sort of nutrient. Here is another transmembrane protein similar to what we looked at before, where it might act as a signaling protein. If another molecule docked right here, it might cast off the piece down there, and it would traverse into the cell in order for that signal to be pushed through. the real piece here is that the membrane is really allows for a living, breathing sort of system in which we allow these very exquisitely controlled processes to specifically deliver and remove any nutrients or waste products, and allow for signaling within from the outside of the cell to the inside, or the inside of the cell to the outside. Let's talk a little bit more about what I was saying about the fluidity on the interior of the membrane. And that fluidity has to be exquisitely controlled. It can't be too fluid, or these membrane proteins would just sort of float out of it. And it can't be not fluid enough. It can't be more solid-like, or these membrane proteins would not be able to float within there or be able to poke through on either side. So a specific level of fluidity is crucial in this. So there's two factors that control the fluidity on the interior of the membrane. And the first would be, here's a close-up of one of those pictures we saw before of the membrane. And notice in there, if you will, we have our lipid bilayer, and on the interior we have these tadpole tails sticking down. These tadpole tails, if you'll remember, were the hydrocarbon tails on our phospholipids. They're all hydrocarbons, so they're hydrophobic, and they just associate to the inside of the membrane where there is no water. Now, the nature of these hydrophobic tails, remember, our fatty acids could be either saturated or unsaturated. Well, these fatty acid tails, since they're derived from fatty acids, can be either saturated or unsaturated. And we saw something interesting way back when we talked about the melting points of fatty acids. That was that saturated fatty acids tend to have a higher melting point, and unsaturated fatty acids tend to have a lower melting point. And that was due to that kinking factor, the bad packing potential of the fatty acid, the unsaturated fatty acids, because they have that kink put into their hydrocarbon tail, whereas the saturated fatty acids had a very good packing potential, and thus they would remain solid for longer up to a higher temperature. The same idea can be indeed applied here when we're not really talking about going between liquid and solid through a melting point, but that saturated fatty acid tails lead to a less fluid-like interior of the membrane. Unsaturated fatty acid tails within here lead to a more fluid membrane. So let's take a look at these phospholipids in particular. When we think of if we look at the top structure here, this phospholipid has a higher percentage or ratio, a greater number of saturated fatty acid tails, whereas this one has a greater number of unsaturated fatty acid tails. So if we look at the comparison here, if we have a membrane which is on the whole has a greater number of saturated hydrocarbon fatty acid tails, those tadpole tails, well, that's going to contribute to a less fluid membrane interior. However, if a membrane has a greater amount of unsaturated fatty acid tails within its constituent phospholipid components, that's going to contribute to a more fluid membrane interior. It's important to see that all of these phospholipids within this membrane are not covalently bonded together. They can be pulled out and new ones can be swapped in, and indeed there are active regulatory processes which monitor the fluid on the interior of a cell membrane, and these processes, if a membrane becomes too fluid, well, they'll pull out the phospholipids that are more unsaturated and insert more phospholipids lipids with saturated fatty acid tails, and that will help to adjust that, that fluidity. So if we had to put this into a nutshell, in a membrane, the greater amount of saturated fatty acid tails means the interior of that membrane is less fluid. Less amount of saturated fatty acid tails means that membrane is more fluid. So that is one way in which the fluidity on the inside of membranes can be exquisitely maintained. There is another component to this, though, another way in which the fluidity of the inner membrane, of the inside of a cellular membrane, can be regulated. And here I've got a couple of pictures of slides we've seen before, but they're highly blown up. And I do that because there's a couple of things within the interiors of these membranes we really haven't looked at yet, or really talked about. And look at the picture over on the left-hand side. What are these things? These sort of golden pieces right there. We see them on both sides of the membrane. And over on the picture on the right, we see these again. Here's this thing right here. Here's another one on the other side. What exactly are these things? Well, these are artistic renditions of the molecule cholesterol. Cholesterol, if we look over far over on the right here, here is the structure of cholesterol. Notice that it is almost entirely hydrocarbon in structure. Entirely hydrocarbon. That is hydrophobic in nature. As we know, hydrocarbon is hydrophobic. But interestingly, on cholesterol, there's one area, that hydroxyl right there. That's OH, that can hydrogen bond. OH can hydrogen bond, water can hydrogen bond. That means that just that tiny section would be hydrophilic. Now, this cholesterol, the body of it, if you will, that whole body, that ring system is rigid. If you remember, it was a rigid system. It was, you were not able to have any free rotation around any of those carbon-carbon bonds within the ring, and they were fused together. So we had this rigid system. Now, this rigid system, when inserted into a fluid cell membrane, and we'll look over here at the picture on the left. Here we see some representations of cholesterol impregnated into a membrane, and this is a rigid structure. This is very similar to reinforcing concrete with rebar. It makes it stronger. This is just an analogy, of course, but the more of these rigid structures that we can insert into that cellular membrane, it would make sense that it would lead to less amount of fluid. Now, interestingly, let's look at how these cholesterol molecules associate within this membrane. Notice we have over here on the picture on the left again, we have a series of them on one side of the membrane and on the other side of the membrane. Look at where, if I can find this, right where these charged head groups are, right on the outside of the membrane, or the other outside of the membrane here. That is where the hydroxyl, let's look over on the right now, that's where that hydrophilic hydroxyl associates, because it is associating with that water on either side. The rest of the cholesterol structure, as we see here, is inside the membrane, where we do not have any water at all. So it spontaneously inserts itself into membranes, or it will stay in there once it is inserted, and regulate the fluidity of that membrane. Essentially, if we look up on the top, a less amount of cholesterol in the membrane means, more cholesterol means less fluid. Less amount of cholesterol means more fluid, because if we're taking away that rebar, that rigid structure, less amount of it is going to make the interior of the membrane more fluid. All right, what the last slide represented was the end of Unit 8, with the end of talking about lipids, and now we're going to move into another type of structure, if you will, under the auspices of biochemistry, and that would be with respect to proteins. proteins are a very, very important substructure within the realm of biochemistry. And indeed, it is such a vast and deep subject that we can really only scratch the surface. So much of biochemistry involves the association of proteins, protein-protein interactions, the sewing of proteins into the fabric of muscle tissue and any other type of tissue we're looking at. It really is a vast type of thing. So as such, we have to really nerf it up somewhat. We're just looking at the generalities of what proteins are, and then we can look at some structure. If we had to break proteins down into two categories, the best way to do that would be to talk about fibrous proteins versus globular proteins. Fibrous versus globular. We will not be talking about fibrous proteins too much, and we'll save most of our discussion towards globular proteins. But just to distinguish them, fibrous proteins tend to be water insoluble. They do not dissolve in water. They have as examples structural proteins such as the proteins in the skin, the hair, fingernails, and that sort of thing, and also movement proteins. These are essentially what muscle protein and that sort of thing. Water insoluble. Now, we are going to be, as I said before, looking much more carefully at what are called globular proteins. These, what distinguishes globular proteins from fibrous proteins is that they are water soluble. They can dissolve in water. Since animal systems and biological systems are primarily made up of water, it is important that certain proteins, most proteins really, can enter the dissolved state. When it can do that, then we can deliver these proteins to cells. Most of biochemistry happens, just as most of organic chemistry happens, in the dissolved state. Examples of globular proteins, and this list is by no means a full rendition, but we could look at biological catalysts. So in organic chemistry, we had catalysts for our reactions. Well, we can't be putting metal catalysts or acid catalysts into our biological systems. So certain globular proteins act as catalysts in chemical reactions. Another example are in the immune response, defensive antibodies. Others are proteins that deliver nutrients or important types of structure to and from the cells. These are transport proteins. Some proteins act as hormones or signaling proteins. These are considered regulatory proteins. And last part of our example we have as nutrient proteins. So it's proteins we can actually gain sustenance from. A big part of this, the biochemistry type of outlook here, or how it's being presented, is looking at the underlying structure of biochemical structures and that sort of thing. We found that starches and disaccharides were based on the monosaccharide. Most lipid structures are based on the fatty acid type structure. Well, in proteins, we're going to be looking at the basic building block of proteins. These are called amino acids. Amino acids. There are 20 separate amino acids that go into the building of all proteins, and we're going to be looking at those in more detail down the road. 20 separate amino acids, but let's look at the structure of an amino acid so we can perhaps derive some of the qualities of why it is considered an amino acid. All amino acids, and this is a very general type of structure, start with a central carbon. That is the alpha carbon. A central carbon, and emanating from that central carbon, we have an amino group, And we also have a carboxylate group. Now, an amino group is always charged at physiological pH. A carboxylic acid is always a carboxylate at physiological pH. However, the fact that they have that these molecules have both an amino group and a carboxylic acid group give the name to amino acids. Amino for the amino group, acids for the carboxylic acid on there. So two of our four bonds emanating from that central alpha carbon are an amino group and a carboxylate group. Almost always there is a lowly hydrogen sticking off of that alpha carbon. And lastly, and most importantly for structure, we have a side chain on any amino acid. That's given as R right here. That R group means because there are 20 different amino acids, there are 20 different R groups associated with them, different identities of that R group. Now, one important feature of amino acids, if you look down the bottom here, is that they are known as a special type of structure known as a Zwitter ion. Zwitter ions. By definition, Zwitter ions possess both a positive and a negative charge on the same molecule, importantly, canceling to neutral. Now, if we look up here at our amino group, we have a single positive charge. If we look at our carboxylate group, we have a single negative charge, and thus that it would be considered a Zwitter ion because a +1 and a -1 cancels to neutral even though those charges still exist within the molecule. The overall charge on the molecule would be considered neutral. But let's take a look at these, most importantly, these R groups that differentiate our 20 different amino acids. Here are the specific structures of all 20 amino acids that comprise protein structure. This is definitely a busy slide, but I've glommed them all together here. We will categorize them in the next lecture, but I've put them all here just to show you that while they're a good component of these amino acids, really, the top half of all of these are exactly the way we saw before. We have our amino group, our carboxylate group, our hydrogen, and the alpha carbon throughout all of these. It is the structures in red below which designate the difference between them in the form of their side chains. That R group, that green R group we saw in the last in the last slide. Now we show what that R group means, and it's given in red on all of these. So if we look on the upper left-hand corner, we can have an R group that's a hydrogen. We could have an R group that's a methyl. We could have an R group that is an isopropyl. And each of these has a different name. It is a different specific amino acid. And we can get more and more complex as we go along, throw in some phenyl groups and some fused rings. And really, we can take some of these and really get complex with them. We will talk about these more and again categorize them in the next lecture. But these different structures, the point is they're all different, and thus they all represent what that R side chain is. Moving forward, we'll look at these, we'll put them into groups to describe the nature of their side chains.